Well, we reached our final lecture on population genetics. So we want to now kind of go over the mechanisms that drive gene pool change. And we're going to spend a lot of time on this because there's formal population genetics classes. You've probably seen some of this in evolution and systematics. But we want to hit some of the highlights, some of them that fit into what we've been talking about. And they certainly, any one of these, you could spend an entire semester uh, dissecting. So we've looked at large random mating populations and seen the effects of when you violate those. Now let's talk a little bit about what these other things are. So one of them is no genetic drift. So first we have to ask, what in the world is genetic drift? And genetic drift are chance changes in allele frequencies. I mentioned again that Darwin recognized this, that things that weren't under natural selection most likely were fluctuating in substance, which means they were under chance changes. Important for us, based on some earlier discussion, is that the smaller the effective size, the greater the effective drift. Okay. Now, drift occurs in all finite populations. Doesn't matter how big or small. The bigger they are, the slower it is, but it occurs, and it always leads to a loss of alleles. It's always taking alleles away, taking mutations out of the system, and it's a chance event. A little chart showing what it might look like. Here's what Hardy-Weinberg predicted, because we have an equilibrium, right? So once you reach Hardy-Weinberg, you could stay there. But if you throw in drift, it's strictly dependent on the effective size. So if you have a large effective size, you'll stay close to the drift that Hardy-Weinberg expected, although eventually over time, if you're long enough, you'll go up or down on the model. If you have a small effective size, you'll bounce even quicker and eventually hit one of these endpoints, either where you have 100% of that allele or none of that allele present in this form. Okay. Let's see if I can find my other page I was looking at. There we go. In this particular page, you can see this is you can go on the web. There's lots of these population simulations. Here we've got a population size of 50, and we've got an initial allele frequency of 0.5. And we're just going to follow some populations over some time. Okay, so here's what would happen if we go 200 generations, right? And you can see that with this population size, very quickly, some of them, this is fixation up here for the P allele, complete loss of the other, but sometimes just by chance it could go down. And the sums can maintain themselves for a long, long time, but ultimately you're going to lose some of those alleles. And if you go down and you say, okay, what about effective population size is very small? Okay, well, you get almost guaranteed that you're not going to go near as long, and very quickly you might lose some of the alleles that you had. This is what happens in conservation settings and things. We say, oh, well, there's plenty of those animals out there. Well, it depends on the effective size. If the effective size is low, then we're losing genetic variation. So even though we might, produce some more of them, get the population numbers back up, if we throw those out there, genetically there may not be much variation left. And so we'll see in a moment what this can lead to. Let's just try one more. Let's just make it 1,000, okay, and see what happens there. Okay, if we make it 1,000, what you see is that this is an effective size about 1,000. You stay much closer to Hardy-Weinberg predicted, right? But ultimately you're going to start to deviate. And eventually you will, over time, now this is a lot of generations, but over time, as this red one's doing down here, you will eventually start to creep down. And this one could actually creep back up. It's chance, right? Although generally, once you get into an up or down trajectory, you tend to get pulled. There's sort of almost a mathematically gravitational pull that pulls you one way or the other. But you can see you've got a very different model than what you had a few moments ago. All right, let me see if I can find my other one here. Let's see, where are we? Okay, so this is what we have there, right? Okay, and we have genetic drift going on as we talked about. Okay, now these are not drift events, but they potentiate a drift. They maximize a drift. Two of those are bottlenecks and founder effects, things that you should know about. A bottleneck is what it sounds like. If you ever looked at a Coke bottle or something, you dumped it over real quick, it takes a while for the material to flow out, right? That's the bottleneck. Well, genetic bottlenecks are this. They're when you get a sudden or sustained reduction in the population size. And what happens in those situations is that you might have some frequency up here of big A being 0.9, little a being a fairly rare allele, 0.1. Okay, it's just found in some numbers. You go through a bottleneck and it's purely chance who makes it. Maybe by chance one of these homozygotes or a het makes it. and then the other one is some other form. Well, look what happens immediately if they assume those are the available individuals to mate in the next generation. Your effective size down here, or excuse me, your numbers down here become completely different. Now, most often what's going to happen is you're going to lose the rare alleles in this situation. This is what happens in conservation settings. But it can be that it actually potentiates the frequency of that allele. We've lost those alleles. Bottleneck has occurred most certainly in cheetahs. You can skin graft between one cheetah and the other. They're so genetically monomorphic. So somewhere back in their history, there was an event which you had a bottleneck, which removed much of the genetic variation. So it's probably a fairly long bottleneck. Same thing's happening conservation-wise with the Florida panther. There's a number of problems that you see there because the population bottlenecked down to a very few individuals. Founder effect, much of the same type of thing. You have maybe a population on a mainland, and that population is at some frequency. The founders won't necessarily carry exactly that frequency, and so you may end up with a change, right? And this is one of the places where we very often see fairly rare genetic issues, genetic diseases in the mainland actually occur in fairly large concentrations on islands. This is a picture of Pitcairn Island, where mutiny on the Bounty. So the mutineers ended up with some Polynesians, they interbred, but one of those individuals from the UK apparently had this brain disease, much like Alzheimer's or Huntington's, and it's become fairly prevalent on the island amongst the individuals that are there. Same thing on Martha's Vineyard in the U.S. The population that established in Martha's Vineyard, this whaling and fishing village, interbred. They were founders from mainlands, right? They had some individuals there that had congenital deafness, and it became fairly widespread or more commonly spread in that population than it was on the mainland, their source population. So you see all sorts of things like that. The Boers that came into South Africa, a number of the Hutterites had high frequency of these fairly rare human diseases, but they showed up in those populations because one or more founders actually were carriers for them or had it. Okay. All right, that's drift, and drift is critical. It's one that you don't get to talk about enough, I think, in general biology and biology things because it's so critical to what goes on. The loss, I mean, just because you can produce these alleles and make them doesn't mean they're going to persist. And then, of course, selection. I just want to point a couple things out about selection. One, it's what we call fitness, right, is one of the definitions. But it has some necessary conditions that are important. They were important for Darwin. They're important for any time you discuss fitness. One is that there's variation, of course, because if you don't have some tall things and short things, then there's nothing to select. So you need variation among individuals. That variation needs to be related to fitness differences. What that means is you need to have a relationship between the trait and some form of ability, such as the ability to attract mates, the fertility of the individuals, the fecundity is how many progeny they can produce, or in the classic, tooth and claw sense, survivorship. Who can survive longer, who can survive more, etc. So there has to be that relationship. And the thing, many people come in and they study selection and they find out there's variation, and then they do some little experiments to show that, yeah, the purple ones do a little better attracting mates than the green ones, and etc. What they often, until recent days, have not had, and what Darwin didn't have was inheritance, right? Just because you're purple and you're green and you might attract more mates, if that's not a genetic trait, I mean, like if it were bloodshot eyes, which you may have from trying to study all this, that's not heritable. That's that's something else. Eye color is heritable, but not that. You have to have inheritance. So there must be, this all must interconnect. The variation must interconnect through fitness differences, which must be under genetic control. Okay. All right. And just because I think it's important, and the book illustrates these, it gives some examples of selection. One is stabilizing selection. Stabilizing selection, if this is a distribution of, say, sizes or weights or something, stabilizing selection is selection that works on the extremes. And what the outcome of that is to produce something that's sort of formed. Now, it doesn't have to be perfectly equal on each side. You could push it over a little bit, but it tends to take the distribution and push it down this way. A good example of that might be human birth weights, right? Before we had artificial wombs, which would allow us to have extremely small children survive. or when we had cesarean sections, which would allow very large ones to survive as well as the mother to survive. The birth weights of actually making it to live birth were fairly tight, okay, within a few, kilograms of size, and that's true for many mammals. So stabilizing selection. The classic selection that you think about in things when we're doing domestication or crop improvement or changing the butterfat in milk or something is directional selection. We start out with a distribution that has some genetic control of that distribution, maybe the amount of butterfat under some partial genetic control, and we want to make more butterfat. We just pick those individuals out here that have the most. That means we limit these, so we push it, and then what we tend to do is then move the mean over, okay? And it will also compress the distribution as well to some point where we can't push it any further. One of the outcomes of any type of directional selection, whether it's natural directional selection or artificial selection, as you might have in domestication or in crop improvement. And then, of course, we have disruptive selection. One of my favorites, kind of cool. Disruptive selection is one where we go against the average, the mean. So it's not good to be in the middle somewhere, right? And what this tends to do is to make a bimodal distribution, okay? Unfortunately, like some undergraduate course grades, okay? But it's a bimodal distribution. So you end up with some big ones and small ones, or some that produce a lot, some that produce very little. So where does this ever happen? Well, it happens in the real world in a number of places. Some of fishes, you'll see this. There's what's called sneaky breeders. They're common in some of the bluegills type species that are found, panfishes that are found along the Gulf Coast of Alabama and Georgia. And what you'll have is the big males will come along and they'll fan, and if you see them, if you drive over those streams where they have the white sand but kind of the dark overlayment of it, you'll see these round puck holes that look like they're little bomb craters. It's where the fish have sit there and finned and made a nest. And the big males will make a nest, and they'll flash, and they'll attract the female to lay eggs. In those populations, you can also have the same species. You have these little sneaker males that are much smaller. They don't build nests, and their job is just simply to fly through, drop off enough sperm that they can fertilize a few eggs and not be, if you're too big, if you're kind of average here, you can't defend a nest. And you can be seen. So you got to be really small or really large. Same thing occurs in some of the morpha butterflies that you see in the tropics. Get a chance to go to Costa Rica places, beautiful blue butterflies. The males will find a good habitat for the female to lay eggs on a plant, and they will protect it and flutter. And then you got these little males, same species, little sneaker males that'll come in and fertilize some of the eggs. So you don't want to be too big. Or excuse me, you want to be big and protect it, or you want to be really, really small. You don't want to be right in the middle because you'll be seen, and these will be, they'll attack and fight you. And you got to be big enough to defend your turf, or you got to be small enough to sneak in and not be a threat. Okay. Now, one of the really important things to tell you about selection is it can be really slow. Okay. Just to give you an example, let's take a recessive lethal. I mean, it just kills you, right? Everybody dies. It gets it, at least in the homozygous condition, right? If you take a selection against a recessive lethal, and you say, okay, I want to just know how many generations it takes to go from some initial frequency to some lower frequency, right? I want to reduce it, okay? Maybe I want to start out with a high frequency. Now, you're not going to find a recessive lethal at this level, but just pretend you did. To cut it in half only takes about 2 generations. But look how many generations it takes to cut it down to a fifth of what it was, right? It takes almost 100 generations. That could be a long time. Let's think about something that's more realistic, okay? If you have one that's around 1%, 0.01, cut it in half, takes 100 generations. Cut it into a tenth of what it was, takes 900 generations, and thousandth of what it was takes thousands of generations. Let's just take this case where we've cut it down from here to here, right? By doing nothing but killing off all the individuals that are that. Okay? Well, if you took humans, we have an average lifespan, reproductive time, rather about 20 years. That's the average. Just maybe a little less, a little more, depending on the population, but 20 years is a good number. Just to make this little change right there, if you got rid of every recessive lethal, if you're successful, would take 18,000 years. Well, you're going to mutate in that time and all sorts of things. I mean, 18,000 years, is much of the time that we can think about of sort of written history or available history for humans. So that's not something that's going to happen. Why is it important? Because these lethals in this case are being carried along in these heterozygotes, right? You can't get to those. So things like genetic counseling, one of the things they do, and certainly with new molecular markers and things, is that we find ways where we can find these carriers, right? Who's a carrier and who's not. If you can remove the carriers and these guys, you change a recessive lethal to a dominant lethal. Dominant lethals only last one generation, right? Now, you can't do all that by genetic counseling, but you can certainly speed this whole process up. So the whole process of trying to figure out who our carriers are is so that we can identify those individuals and recommend that they don't reproduce or try to correct the situation or something else. The idea being that we're never going to get rid of genetic diseases. And think about this, once you get really low frequency anyway, where it's a really rare genetic disease, it's almost nobody actually has it like this. It's always in the carriers. If you can't find the carriers, you're going to keep that particular mutation going on further and further. And to add to all this, if it's not a recessive lethal, it's some less amount of selection. It's even slower than this. So the point of all this is that selection is really, really slow process. Okay, it may be strong and continuous, but very often it's a very, very slow process. And meaning that drift can often move you around a lot faster than selection can. Okay? And just a couple of others, mutation, just so you know, for obviously mutation is the only source of new genetic variation, right? We don't get new variation from selection. We don't get new variation from drift or anyplace else. In fact, we usually lose it through both of those. The only place we're going to get a new mutant, a new thing, is through mutation. So it's critical because it puts in that variation that we need that then can be used in sexual reproduction and independent assortment and all that. Okay. The actual assumption in the Hardy-Weinberg model is that the mutation rate is equal. Forward and backward are actually equal. So once you mutate to this, you mutate back at about the same rate. That's not going to happen. So it has an effect, and people do have incorporated mutational rate changes into these Hardy-Weinberg models. Now, for most of us studying mammals or studying plants or whatever, the generation time is so long that mutation doesn't play as great of a role. It's actually, while it is the ultimate source, it's not one of the big factors that we think about. Okay, one of them that we do think about though is migration. Now we've already covered this a little bit. We talked about founder effect and sort of those things, right? Well, that's migration, right? So migration is what happens as far as movement between populations. And it's the basic idea that populations exist, they may be inbreeding or whatever, and then you have this movements that go on in between them, and you can calculate up for those, okay? And one thing that's really, really turns out from the mathematics of all this, and it's really interesting about migration, you don't need much effective migration to keep populations genetically relatively equal, okay? Theoretically, and it turns out to be slightly more than this, but theoretically, you only need one effective migrant per generation, say, to keep these two populations at the same allele frequencies. Okay? Even under fairly strong selection and all sorts of other things, it doesn't take a huge amount of migration to actually keep these things pulled back into some form. It's a really interesting outcome, and it may take more than one, but that's the mathematical bottom line of where it is. So migration can be really important and really fun to study, and migrational patterns and who leaves genes where and all that. Really interesting area of population genetics, and most certainly now with all the ability to travel throughout the world, migration and gene flow relative to diseases and pathogens is critically important in geographic medicine. Okay, all right. So now we've taken gene pools. We've decided that they can change over time. The predictions, the straight down prediction is the Hardy-Weinberg equation, which predicts an equilibrium. And we can see that equilibrium can be broken up by a number of factors that all contribute. And these are all mediated under effective population size, right? So drift will go way up if population size is low. Selection may go up if it's small, or big, either direction. Okay, it can have an effect. Okay, populations get really huge, then they're going to migrate and move out. All right. And above all this, of course, is environment. Environment is controlling population size, mutation rates, drift, whether or not you can or don't migrate, that sort of thing. of course, studying all this, really we talk about population genetics, but in reality, what we're studying, these particular mechanisms of gene pool change, are the mechanisms of evolution. So population geneticists are really interested in getting down, uncovering the rocks about and looking to see what drives the evolutionary process. Because you can define evolution as a change in gene pools over time, right? And if you do, what's changing them? Well, it's one of these mechanisms in relationship to environment. This has led to a really interesting fight between some groups. There are these neutralists. Neutralists are those individuals that believe that most of the changes in the genetic code are so small that you really can't detect them by natural selection, right? Natural selection needs these fitness differences, right? Like the ability to take mates, or the ability to put out more offspring, or for your offspring to survive better, fecundity, or whatever. Well, if we've seen all these mutations, right? We've seen that there's millions and millions of places that you can mutate any genome, and certainly hundreds of thousands of ways that any one gene can be mutated. And what's the chance that it's going to change whether I have 2 kids and you have 1, right? That's going to likely. So what they argue is they're so small that chance changes really drive it. What this means is organisms aren't particularly well adapted necessarily, right? We don't, this concept of, oh, it's perfectly adapted to be out there standing around in the cold or this or that. It might be, but the way it got in there may in fact be more driven by chance than it was by selection. And of course, this is diametrically opposed by neo-Darwinian selectionists who say, oh, poo-poo. No, drift is kind of going on, but selection is driving it. Things that are picking it, making you better, persisting, and all that. Organisms become more and more and more adapted. Okay? Led to some really interesting models and things about it. And this was a huge fight that went on back in the 1970s through the 80s and selectionists. Most of you listening to this are trained as neo-Darwinian selectionists. Okay, that's what you've heard. That's what you hear on TV and how things work and all this other things. But the reality is, it's somewhere in between these goals. In fact, in the modern world, okay, Darwin talked about variation, inheritance, and selection. The neo-Darwinianists pulled in mutation, and of course Mendel got involved. That's where our population genetics started to grow, and then we had all these things. But the real world now is over on this red side. It's all of that, okay, except now we know things about epigenetics, which we've talked about, right? There's this idea of plasticity, there's natural variation, there's genomic wide evolution, there's the ability of this species to map its genes on the thing that it eats or the thing that it lives on or whatever. There's all this sort of variation out there. So just being a neo-Darwinianist and thinking through that everything's natural selection and blah, blah, blah is a really, a naive way to approach the system. We're now moving along to a very complicated and much more informed way of thinking about it. Okay. All right, that concludes our population genetics. Hope you've learned a little bit from that. I suggest you look through the chapters of the book. Again, we skip a huge amount of the material there. You can concentrate on the lecture notes for the test and all that. It's pretty much going to pull from here. I just wanted to give you an overview and try to highlight some of the things that you might not see in a sort of a classical course that talks about population genetics or about evolutionary process.